Everyone. Welcome back to the series of videos in which I go through the AP progress checks for each unit and I just break down the FRQ questions. Um, and again, hopefully you guys are doing these on your own first and then checking your answers here. It's probably the best way to go about it. Okay, so now we're going to dive into the Unit 5 progress check. AP Unit 5 is Heredity, which pretty much corresponds to our chapters 13, 14, and 15, which is kind of, meiosis and chromosomal genetics, right? So kind of earlier in the semester, but we did this in January and February, so it should be a little bit more recent than some of the other stuff we've talked about. Okay, so let's take a look at question 1. So the sex of an organism is typically determined genetically, but environmental factors can also play a role. Scientists examine the role of genes and environmental temperature in the sex determination of a lizard species. Individuals with a ZW pair of chromosomes are always female, while individuals with a ZZ pair of chromosomes can be either male, ZZM, or female, ZZF. Scientists mated ZZWF or ZZF females with ZZM males and incubated the eggs, produced in 20 clutches at temperatures ranging from 23 degrees Celsius to 36 degrees Celsius. When the young hatched, the scientists determined their sexes. Among the eggs that did not hatch, the mortality of embryonic lizards was approximately the same for both males and females. Okay, so let's take a look at what they want. Okay, if a particular gene is located on the Z chromosome of this lizard species, describe why a lizard with a ZW genotype has a greater possibility of expressing the recessive phenotype for the trait than a lizard with the ZZ genotype does. Okay, well, this is exactly like X-linked disorders, like my very own color blindness. So it's the idea is that a lizard with a ZW genotype, which would be a female in this case, is what we call hemizygous, right? Is hemizygous, meaning that there's only one copy of the gene, right? So the idea is if this Z chromosome, right, if this gene, if this is recessive, then the organism is recessive because that's the only copy they have. Whereas a ZZ male or a ZZ female, doesn't really matter, would still have two copies of this allele. So even if one is recessive, the other one could be the dominant. Right. So basically, one recessive allele is all it takes to show in the ZW individual, all it takes to show the recessive phenotype. Just like, I am colorblind, I only have one copy of the color vision allele because I got my other chromosome is a Y chromosome. Okay. All right. So let's take a look at B. Ooh, good stuff here. So using the template, construct an appropriately labeled graph, including error bars. Oh yeah. To represent the data in table 1. So based on the data, compare the ZZM times ZZWF crosses and the ZZM times ZZW or ZZF crosses to determine whether there's a significant difference between the two crosses in the fraction of female progeny produced at 24, 32, and 36 Celsius. Okay, so I will be the first to admit that this graph is kind of a doozy, but if you just take it one step at a time, it's actually not that bad. So first, I just kind of took the data table and I just said, okay, these are the temperatures that I'm supposed to be looking at. And then I just sort of set that up on my X or my horizontal axis, 24, 32, 36. And again, I think you could probably represent this data a couple of different ways. I'm going to do a bar graph on this one, just because it seemed to me to be the easiest way to do it. I'm sure you could do it in a line graph if you wanted to, as long as you had two separate lines. Or let's see, I guess you'd have to, yeah. Okay, So you'd have a line for these crosses and a line for those crosses. But I don't know. I feel like it's easier to represent in a bar graph. And then on my y or my vertical axis, right, I've got fraction of female progeny. So really even number, like so 3 temperature things, 3 dark lines here, and then 25, 50, 75, 1.0. So easy scale to work out. And so these are, not the neatest. It's not the neatest graph I've ever made, but these tools on uh, Active Inspire or whatever my little program that I've got here are not the best for free drawing. So, but you can see what I did here. I kind of I plotted my data, right? So, and then this I used to make my error bars, right? So we've done this a bunch of times in class. This is going to be 2, and this is standard error of the mean. Um, Again, if I were to make up a way to abbreviate that for teenagers, I probably would have done it differently, but whatever. Um, so 0.5 plus or minus 2. So when I draw my error bars, I make it go from approximately 0.52 down to 0.48, right? Because it's going to be this way on either side. That's what this plus minus means. And then here there's this, it's a 0, so there's no error there. And then you can see I did, 0.3 on either side, right? 0.2 on either side. And then here, no standard error. The mean those data points are identical. So that's part 1 of question B. Oops. And then secondly, what they want us to do is they want us to say, is there a significant difference between the 2 crosses at 24, 32, and 36? So what it looks to me is that, yeah, there is no overlap of error bars here or here, right? There's nothing there. So I am going to say significant difference. Significant difference at 24 and 32. And then at 36, though, at 36, no significant difference. Okay, so at 36, no significant difference, right? Because the data points are the same. All right, so let's take a look at what the answer key here. So, okay, 1 copy of the gene with the ZW, while the lizard with the ZZ has 2 copies, one of which might be the dominant allele. Fair enough. Sketched bars are again, this correctly plotted line graph. If it says bars here, I'm thinking that you can definitely get all 3 points with a bar graph as long as all your stuff is there. And then when you're analyzing data, okay, so significant differences at 24 or 32 due to no overlap of the error bars. And then identical data for the 2 crosses at 36, so no significant difference. Okay, so let's look at part C. So for each cross, describe whether genetics or temperature determines the sex of progeny produced from eggs incubated at 35. Right, it's going to be right here. For each cross, describe whether genetics or temperature determines the sex of progeny produced from eggs incubated at 24. So we're really comparing this here. Okay, so yeah, so all we're doing is we're taking a look at this data versus that data. Okay, so all right, so what I did just to kind of clear things up a little bit is I kind of drew a couple of Punnett squares, and the way I did that is I just basically broke down, okay, if we got a ZZ and a ZW, I just, broke that. These are the gametes, right, that it could produce, and here's how they would combine. Okay, so looking here, so at 35, you would expect a 50-50 ratio because these are always male, these are always female. So at 35, though, that's not what you see. You see 93% of the male or of the offspring develop as female. So what that tells me is that at 24 or sorry at 35 degrees, it's going to be each cross is temperature, right? So both crosses are okay. Actually, sorry. Let's go one cross at a time. It's probably going to be easier. Okay. So looking at this, um, sorry, you can probably hear like my kids screaming upstairs and stuff like that. So working from home, good times. So okay, so looking at this, so at this first cross, right, that I set up here, you would expect this. So at 35 degrees, the ZZM, right, the ZZM, right, times ZW female, right, then at 35 degrees, that's environment. But at 24 degrees, this cross does actually look like what I would expect from my Punnett square, right? It's 0.5, right up the board. So it looks to me like, at temperatures of 30 degrees or under, 32 degrees or under, this is determined by genetics, right? So this one, this one here is going to be genetics. But this other one, ZZM times ZZF, looks like to me, honestly, there's no, because you got to remember that ZZ can be either male or female depending on temperature. So what it looks like to me is that there, it doesn't matter at all what the Punnett square is. It's just like at lower temperatures, they're all male. At higher temperatures, they're all female. And if you graphed it, which I did not do, but if you did, you would just see like a straight line going in from male to female. Like at 33 degrees, they're half and half. So looking here, that's going to be environment. So in both cases for this cross, I'm going to go ahead and say that it's the environment only. Oops. Okay, cool. And then, so last part of the question here. Predict the effect of increasing global temperature on the continued presence of the W chromosome in this species of lizard. Scientists claim that in this species, the W chromosome is unimportant in sex determination. Instead, proteins that are encoded by a gene or genes on the Z chromosome and that are maximally expressed only at certain temperatures are responsible for determining whether embryos will develop as females or males. Use data to support this claim. Well, okay, so predict the effect of increasing global temperatures. Well, so you can see that basically the W doesn't really play a role in sex determination at higher, especially at higher temperatures. So as the Earth gets warmer, right? So as the climate gets warmer, what you'll probably see is more ZZ females out there, right? You see more ZZ females. So it's possible that the ZW would disappear, just either randomly through genetic drift, or I mean that's probably the most likely scenario, because it probably wouldn't ever be disadvantageous, but it might disappear randomly through genetic drift, or it would certainly decrease in frequency. And then proteins that are encoded by a gene or genes on the Z chromosome um, are responsible. Okay, yeah. I mean, you can definitely see that in the data here for sure. because it really looks like you're only male. You're only male if you have uh, pretty much two copies of the Z chromosome, and it's at lower temperatures. So basically, it's probably the idea. I would say that this to be male, or sorry, there's probably genes that are expressed that give female characteristics, um, and or the genes that express for male characteristics are inhibited at higher temperatures and only express at lower temperatures. So let's see what they were looking for here. So okay, so for C, both crosses, the temperature determines all embryos will develop as females if the eggs are incubated at 35, and then uh, the response indicates that for this cross, about half. Okay, and that contrasts with this, in which temperature determines the sex, because none of the embryos will develop as females. Eggs are incubated at this low temperature. Okay, so then D, W chromosome may disappear from populations, especially those in warmer habitats, right? Because there will be more ZZF females. Um, and the response indicates that embryos can develop as females without the W chromosome, and embryos can only develop as males in the presence of two Z chromosomes and only at certain temperatures. Since one Z chromosome is insufficient for male development, it is likely that a sufficient amount of the Z chromosome encoded protein is only produced from two copies of the genes on this chromosome and at lower temperatures. Okay, so cool. Like I said, this was kind of a longer one because this was rather involved. So I will make another video and post it here shortly about question 2 of this progress check. So thanks for watching, and